Well, thank you so much for the invitation to speak. And I'm just so sorry that I can't attend in person. I see the speaker lineup and all the interesting talks that are being offered. And I know that I'm missing a very special event, but I'm happy to participate even in this way. So today I'll be talking about how environments of ancestors influence traits in the present in the model system of Arabidopsis thaliana. So my research program has two main components. The first is how genes and environments interact to produce ecologically important traits. And the second is how genetically based traits influence the environments that organisms experience. And to the evolutionary ecologists in the audience, you can see that this reciprocal interaction between these two components has the potential to lead to some interesting dynamics and feedbacks. But for today, I'll focus on the first component here, but I'll do so within the reference to one of these traits that influences the environments that organisms experience, namely seed germination. So seed germination is genetically controlled, but it is also strongly mediated by seasonal environmental factors. On the other hand, the timing of seed germination determines the post-germination environment that plants experience and even the environment that they experience throughout their life. For example, the seed that germinates in autumn will experience cool, wet conditions and not much competition, but a seed that postpones germination until the springtime will experience warmer, drier conditions and a full vegetation canopy to compete with. So seed germination really is one of those traits that can determine the environments that the organism experiences. But back to the main focus of this talk, here's the dilemma. Genes or DNA determine the phenotypes that natural selection acts on, and that DNA is stably inherited across generations. So DNA sequence, therefore, reflects natural selection that occurred in the past. But environments can change even within the lifetime of a single individual. Therefore, it makes a lot of sense that the environment, too, can regulate phenotypic expression to produce phenotypes that are adaptive in the environments that organisms are experiencing in that moment, not just environments that exerted selection in the past. So we call this phenotypic plasticity, the ability of a single genotype, as determined by this line here, to alter its phenotype in response to the environment. And here's a classic example of phenotypic plasticity in plants, that of the shade avoidance response, when plants that are grown in full light are shorter and have more branches, but those that are grown under a vegetation canopy elongate, get taller, presumably to overtop their neighbors and capture light that way. Here's another example of phenotypic plasticity, that of seed dormancy. So dormant seeds do not germinate, but non-dormant seeds do. So seeds with low dormancy have high germination potential, and those with high dormancy have low germination potential. And seed dormancy is highly responsive to temperature and other environmental factors. So we have inherited DNA sequence that reflects adaptation to past environments. Phenotypic plasticity modifies phenotypes in response to present environments. But what predicts environments of the near future? Well, environments of the recent past, that is, environments of recent ancestors, may well predict the environments in the near future. So for instance, a new seedling germinating early in the season may not have any neighbors at all yet. There's no cue of future competition in the present environment. But a canopy might well develop later, and competition might be very intense later in life. So what can cue that future environment of competition? Well, the maternal environment can, because that is the environment that the maternal parent experienced during seed maturation. So the mother's environment as an adult can actually predict the offspring's future environment even better than the young offspring's own environment. So this brings us into the realm of parental environmental effects, in which the environments of parents influence the phenotypes of their offspring. And we have many examples of these parental effects across diverse taxa, from plants, insects, mammals, even human beings. And Sonia Salton's book here discusses that at length. So today I'll be giving you some of our results that document how the environments of the past and of the present influence life history traits, namely seed germination, And for the second part of the talk, I'll discuss what we know about some potential mechanisms that control these ancestral environmental effects. So I'll start off with one big experiment that was conducted by my former postdoc, Mariano Alvarez, and my former technician, Andrew Bly. And the object of this experiment was to answer these two questions. Simply, how long do environmental effects persist within and across generations? And how do past and present environments interact to influence phenotypes? That is, our ancestral environments, do they have stable effects on phenotypes regardless of subsequent environmental variation? Or do environments in both generations interact? So to do this, we implemented two diurnal thermal regimes that we grew our plants in, a low thermal regime and a high one. And we imposed those thermal regimes for 3 successive generations to produce every combination of those thermal regimes across these 3 generations. And then we measured traits of the plants in this generation in 6 different genotypes. So we could also assess whether or not there was genetic variation for responses to these environments. So this experimental design allows us specifically to look at the response just to the individual's own environment, to the parental environment, and to the grandparental environment with 2 different starting conditions. So the first question is simply, how long do environmental effects persist? And one hypothesis we might have is that these environmental effects might gradually just dissipate over time across successive generations, such that if we look at the magnitude of the response to the environment, we see that it is very strong when the individual is responding to its own environment. The response to the parent environment is somewhat weaker, and the response to the grandparent environment is weaker yet. Now, this hypothesis would be consistent for a mechanism in which environmental effects are gradually diluted over time through cell divisions, for example. So this is what we found. We did not find evidence of the gradual dilution of environmental effects over time. In fact, for seed germination, the response to the parent environment was almost always stronger than the response to the seed's own environment. And this was true for both of these starting conditions. So each one of these lines is showing you the mean response to the environment for each of these different phenotypes. So the response to past environments can actually be stronger than response to the present environment. Not only that, response to environments in different generations can differ not only in magnitude, but even in direction. So if you look at this blue genotype here, or this yellow one, we see that warm conditions experienced by the seed themselves reduce germination, but warm conditions in the parents increase germination. So there's clearly, it's not a dissipation of environmental effects over time. These are 2 distinct responses to the environments in different generations. And we also see that the genotypes differ in the relative strength of their environmental effects, as shown by these crossing reaction norms here, indicating that there is genetic variation for how different genotypes respond to present and past environments. So the second question is, how do past and present environments interact to produce phenotypes? And I'm rephrasing this somewhat to ask, do ancestral environments modify responses to present environments? In particular, do they reinforce those responses or do they antagonize them? So here's a hypothetical scenario in which successive generations of a single environment can intensify a response. From a switch to cool to warm environment, we see an increase in the phenotype, and with increasing generations of that same environment, we see an intensification of that response. On the other hand, an antagonizing response would look like this. Again, a switch from cool to warm would increase the phenotype, but successive generations at that same environment would cause a dampening of that. And that can happen in either direction, but a reinforcing effect would be monotonic, antagonizing. So for seed germination, with regard to successive generations of warm, in almost every example, we found that the ancestral environment actually antagonized the response to the present environment. From a switch from the low thermal regime to the high usually resulted in a decrease in germination, but ancestral generations at warm temperature antagonized that response. With regard to successive generations of cold, we see that the genotypes differed in whether the ancestral environments antagonize that response, as shown in this yellow one, or reinforce that response, as shown in this blue one. So to summarize the results from the phenotype of seed germination, we found that present environments alter phenotypes, unsurprisingly. But ancestral environmental effects also influence those phenotypes. Even grandparental environments can influence traits in the present. We also saw that there was really no pattern of the gradual dissipation of environmental effects over time. They do not dissipate, but responses to past environments can even be stronger than responses to present environments. With regard to the second question, we found that ancestral environments can either intensify or antagonize responses to present environments depending on the genotype, which leads to the third question, the third observation, that genotypes differ both in how long those environmental effects last and in how the environments in two different generations interact, indicating that there is evolutionary potential, that there is potential for the persistence and the environmental contingency of these responses to evolve. So, excuse me. Now I'll talk about some of the potential mechanisms of ancestral environmental effects. And starting with the very same experiment in which we not only measured seed germination, but we also measured gene expression in those seeds, whole genome-wide gene expression in one of those, and I'll talk about the gene expression results in one of those ecotypes, the Colombian. So one hypothesis, if we were to expect that at the level of gene expression, genes would be responding more to the present environment than the past, we might expect this pattern such that many genes are responding to the environment of the present. A subset of those might be responding to the parent environment, and a subset of those might be responding to the grandparent environment. Well, given that we did not see that for the phenotype itself, it should not surprise you that we did not see that at the level of gene expression. Instead, we saw this. Many genes are responding to the environment regardless of the generation in which they experience that environment. But more interestingly, some genes responded just to the environment of the present, while other genes responded just to the environment of the past, and many of them were responding to particular combinations of environments in the past and present. So clearly, responses to ancestral environment are not simply diluted responses over time. They appear to have distinct genetic bases involving distinct genes. Now, when we look at the magnitude of the response to the grandparent environment and the offspring environment, we do not see any evidence that the magnitude or the strength of the response to present environments is stronger. When we look at either the absolute value of the response or the degree of methylation, in fact, we see the opposite. More genes are methylated more in response to the grandparent environment than the offspring environment. However, if we look only at the subset of genes that were responding both to the grandparent and the offspring environment, we do see evidence that the magnitude of response to the offspring environment is stronger than the grandparent environment. And this really is the only example in which we find evidence of a weakening of the effect of past environments over time. One other thing that we observe is that most of the genes that are responding to the ancestral environment, their expression is also highly contingent on the present environment. There are very few genes in which are responding to the ancestral environment in a way that is stable and unconditioned on environmental variation subsequently. And finally, if we look at the gene ontology categories of genes that are responding to the grandparent environment, the offspring environment, or those genes that are integrating their responses across generations, we do see some evidence of functional differentiation. Unsurprisingly, many of these genes are categorized in the category of responses to abiotic or biotic stimuli. But more interestingly, when we look at those genes that are specifically responding to the grandparent environment, we see a lot of genes involved in mRNA processing, whereas those that are responding to the offspring environment tend to be involved more in catabolism and metabolism. So there is some functional differentiation as well. So to summarize our results on gene expression, we did not see much evidence that at the level of gene expression, responses to environments in the present are stronger than those to the past, either in the pool of genes that were responding or in the magnitude of their response. We only saw that when we looked at the subset of genes that were responding both to the grandparent and the offspring environment. We also found that most genes that are responsive to ancestral environments are highly contingent on the present environment as well. But what we can say is that responses to present and past environments are genetically distinct. They involve different genes, and those genes do have different functions. So we see that gene expression does respond to ancestral environments. But the next question is, what is regulating that gene expression? Well, at this point, I can say we don't know, but one hypothesis is that it is DNA methylation. So DNA methylation is the addition of a methyl group to a cytosine residue in a DNA sequence. And in plants, DNA methylation is associated with the repression of gene expression and the suppression of mobilization of transposable elements. Methylation has been shown to be inherited mitotically, that is, the methylation mark can be copied along with the DNA strand during mitotic cell divisions, and it has even been shown to be inherited across organismal generations meiotically. So DNA methylation actually is a pretty intriguing mechanism whereby an environmentally induced change in methylation status might actually be able to be transmitted directly from parents to offspring. So my former graduate student, Brittany Morgan, was interested in testing whether or not DNA methylation is involved in parental environmental effects. And she was interested in the parental effect caused by vegetation shade. So Brittany manipulated the canopy environments of both the parent and offspring generation. She grew the parents in either full light or shade, took those seeds and then exposed those seeds to either full light or shade. And then she measured seed germination as one of her phenotypes. She also measured genome-wide, she also manipulated genome-wide methylation in both parent and offspring generations separately. And she was able to do this with a chemical demethylating agent, 5-azacitidine. But before that, she was able to verify that the chemical demethylation very closely phenotypically resembled genetic disruption of a major methyltransferase in Arabidopsis-1, so that any phenotypic results that she saw, she was confident they were caused by the demethylating action of 5-azacitidine as opposed to any toxic side effects. And she imposed that demethylation treatment at a very early life stage, both in the parent and in the offspring. So this is what she found, and let's look at the middle panel first. This is the panel that is showing proportion of seed germination for seeds that themselves experienced full light or canopy shade, and for seeds that were matured under full light or canopy shade. And what you can see is that there's an interesting response such that if the seeds themselves experienced full light, they were induced to germinate more. But if they were matured under full light, they were induced to germinate less. So the very same cue had opposite effects on seed germination, depending on whether the seeds themselves experienced it or the mothers did. So now let's look at what happens when the parents were demethylated. And you can see that in that case, the parental effect on seed germination was completely obliterated, although seeds were still able to respond to their own environment. When offspring were demethylated, we see that the ability to respond to the offspring's own environment was obliterated, but the parental effect persisted. So what this means is that methylation in parents appear to regulate the parental effect on seed germination, whereas methylation in offspring appear to regulate a seed's ability to respond to their own environment. So clearly DNA methylation is involved in these parental effects. And the next question is whether or not methylation itself is inherited across organismal generations. So to address that question, we did another experiment. This one, again, led by Mariano Alvarez and another former postdoc of mine, Gabriela Ofi. And for this experiment, we manipulated just the parental environment, another thermal manipulation. And in this case, it was a chilling treatment, a prolonged chilling treatment that is a cue of the passage of winter that many plants need in order to be induced to flower. So parents were either chilled or not chilled, and then we collected some of their reproductive tissue. We allowed those individuals to set seeds, and then we collected tissue from their progeny at the seed stage. And we conducted whole genome bisulfite sequencing on both of these tissue samples. And we were especially interested to test whether or not those loci that responded to the parental chilling in the parent generation were the same loci that responded to that chilling treatment in the progeny generation. And it's those loci that would be consistent with the hypothesis that methylation itself is being transmitted across generations. So I'll plot it in this way, where this axis is representing demethylation or methylation in the parents, and this axis is representing demethylation or methylation in the progeny. Genes, loci that responded just in the parental generation would show up along here. Loci that responded just in the progeny generation would show up here. But it's on this diagonal that we would expect that if there were a methylation change in the parents that was directly inherited by progeny, we would expect them to line up on this diagonal here. So this is what we found. Not a very strong signature that methylation marks are being directly inherited from parents to offspring. Yes, we do see some candidates in which differential methylation in the parents are also genes that were differentially methylated in parents are also differentially methylated in progeny in the same way, which is consistent for direct inheritance, but that is by no means a general pattern that we see. Instead, we see that many genes are responding just in the parent generation, and sometimes different genes are responding in the progeny generation. Some genes are even responding in the opposite direction in parents and progeny. The DNA methylation appears to be involved in parental environmental effects, but it does not appear to be directly inherited very often at all. That means that something else must be transmitted from parents to offspring, and that something else, in turn, is influencing the patterns of methylation in offspring. So the final question is, what is it that's actually being transmitted from parents to offspring? Well, at this point, I can't tell you what it is. We don't know, but we are investigating. I showed you earlier in the talk, we have evidence that gene expression itself is responsive to the environments in different generations. Now we're putting it together with DNA methylation data from that same experiment, as well as small RNA pools to see if we can find any concordance between loci that show differential gene expression and differential methylation. And because we're working with Arabidopsis, we can use some information on which genes are regulated through pathways involving small RNAs to see if we can get further understanding into the gene expression patterns that we found. But we do know that many other things can be transmitted from parents to offspring, including histone modifications or any kind of gene products, hormones, proteins, many different kinds of substrates, anything in the cytoplasm, including organelles. We know many things other than DNA are transmitted from parents to offspring. And so one interesting question is, which one of these mechanisms, which of them have any mechanisms for their own self-replication or perpetuation across even more than one generation? And at this point, I just want to toss out one other thing that might be inherited from parents to offspring, and that is the environment. Perhaps it's not the epigenetic changes themselves that are being inherited, Perhaps it's the environment that induces or reinduces those epigenetic changes that is responsible for the perpetuation of epigenetic changes across multiple generations. So we're continuing to explore that idea. So in conclusion, we've seen that responses to present and past environments have distinct genetic bases. They involve separate genes with separate functions. They also exhibit genetic variation, indicating that these responses can evolve if they are the subject of natural selection. We've also seen that DNA methylation in both parents and offspring is involved in ancestral environmental effects, but that methylation is not necessarily directly inherited as a primary mechanism for the transmission of these environmental effects. Many things could be inherited from parents to offspring, including environments. And I'm just going to toss out there the possibility that the inheritance of environments themselves can influence the perpetuation of epigenetic changes across multiple. So with that, I would like to thank the many members of my lab who contributed to these studies and my funding sources.